Hello, everyone. I'm Denise Marino Mitchell, and I just wrapped up my master's thesis in Lou Santiago's Ecophysiology Lab at UC Riverside, and I'm happy to share with you my thesis today: coordination of drought and heat tolerance in woody species across ecosystems. Now, when one studies drought tolerance, it is helpful also to examine heat tolerance as well, because high heat accelerates drought. But, however, a topic search of all years in databases on the Web of Science revealed that research on combined drought and heat stress is receiving little attention relative to the papers published on the single stresses alone of drought and heat. But studying drought and heat stress combination is essential, because we cannot extrapolate responses to stress combinations simply by the addition of the responses to the two single stresses. There exists a synergistic effect given the nature of the potential interactions, both in climate and physiological regulation. So while both high heat and aridity are environmental extremes that can negatively affect the productivity of plants, many species have traits that mediate their heat and drought risk. And one well-documented avoidance strategy is the drought deciduous growth habit, shedding leaves during the dry period and then growing new leaves when the conditions become more favorable. And leaves of deciduous species undergo greater leaf hydraulic dysfunction to maximize their carbon uptake, yet they are shed and they're replaced annually. Whereas the evergreen species hold on to their leaves, so there's a greater necessity to preserve the vasculature. However, for woody species that do not avoid these stresses by losing their leaves, how can they endure combined heat and drought? And so surviving the stress does not come without a cost. The plant may need to invest in effective osmotic control within the leaf cells by adjusting their leaf turgor loss point, which is the leaf water potential, at which the mesophyll cells lose turgor. So during drought periods, this is accomplished through the accumulation of cell solutes, and such an adjustment results in the lowering of turgor loss point and contributes to the drought tolerance of vegetation worldwide. Now, plants also exhibit unique thermoregulatory traits, both morphological and physiological, and that influence leaf temperature and decouple them from the ambient air temperature. For example, leaves with a low leaf area and narrow leaves are generally associated with a thin boundary layer, and they can cool down more quickly with evaporative cooling relative to leaves with greater leaf width and larger leaf boundary layers. Leaf width is the most important leaf morphological driver of species differences in the leaf to air temperature differential, as is overall leaf size, with wide and large leaves warming up more than the narrow and small ones under similar conditions. So in addition, leaves displaying epidermal modifications such as hairs and cuticular waxes can moderate the incoming radiation, and light-colored leaves can reduce absorptivity. Leaves that roll, have a vertical angle, or maintain a parallel orientation to the incoming radiation can also moderate temperature. Now, in Southern California, there are distinct ecosystems that occur within a 100-kilometer radius. The Mediterranean type shrubland is actually one of 5 such uniquely biodiverse regions across the globe, and it's characterized by warm and dry summers, and it's coupled with mild, wet winters. And this unique geographical phenomenon provides for a wealth of biodiversity and floristic complexity, known as the coastal sage scrub and the chaparral. Now, if we go inland from the coast and we lose the moderating effects of the marine layer, the range of air temperature broadens significantly, and this gives rise to our serene Mojave Desert. And if we ascend in elevation to approximately 1,770 meters, we witness another shift in the resident flora within the Idlewild mixed evergreen forest with temperate summers and cold winters. Now, these three distinct ecosystems provide an ideal framework for relative heat and drought comparison studies, as there exists a significant gradient in both the mean annual precipitation and the mean annual temperature. As you see here in our study system, comparing the mean annual precipitation with the three ecosystems and also the comparison of the mean annual temperature between the three ecosystems. Now, if we look at the site map, interestingly enough, you will notice that latitudinally, these three field sites are very similar. And so therefore, the gradient that we just observed in mean annual temperature and mean annual precipitation is mostly due to the coastal effects on the shrubland and the elevation change in the forest. Now, drought tolerance and heat tolerance were quantified on a wide range of taxa, including 21 woody species spanning 14 families. And our seasonality defined our experimental design. So our wet season measurements were taken in February of 2020 and 2021, and it served as our control. And in the dry season measurements were taken in July of 2020, and that provided our stress data. Now, we are all somewhat familiar with the concept of turgor loss, even if it's just coming home after a long vacation, if we can take vacations again, to see this site, a wilted houseplant, upon arriving home. And so across plant species, drought tolerance is strongly correlated with two physiological traits: the leaf water potential at wilting, which is your pi sub TLP, and the cell solute potential at full hydration, which is your pi sub naught. Now, a lower, more negative value for turgor loss point would indicate higher drought tolerance, and conversely, a higher, less negative value would indicate a relatively higher vulnerability to drought. And so with respect to heat tolerance, at some point of extreme heat, the photochemistry becomes damaged, and this causes a decrease in the photosynthetic efficiency. But at what point in the photosynthesis cascade does this occur? So early photochemistry research found that the electron transport, which is driven by photosystem 2, is the most heat-sensitive process in green leaves, and it is the leading limiting step of photosynthesis at high temperature. So heat tolerance determined using fluorimetry provides an insight into the effect of high temperature on the function of photosystem 2. And so accordingly, we chose chlorophyll A fluorescence as a proxy for photosynthetic efficiency in our study. And the parameter for relative heat tolerance was assigned the temperature at 50% loss of maximum quantum yield of photosystem 2, or T sub 50. And so the higher the value for the T50, the higher the relative heat tolerance. Now, we were interested in identifying correlations of drought and heat tolerance within single ecosystems across species, but also across the ecosystems, and that have distinctly different precipitation and temperature regimes. And then lastly, also within distinct leaf growth habits. So let's take a look at a couple of highlighted results from our research. So this first slide of data that you're looking at is going to be the range of turgor loss point on the stratified by biome on the left, and the range of our T50 stratified by biome on the right. And if we start with the plot on the left, what we see is we observe the widest range of turgor loss point values in the desert during the dry season, and a substantially narrow range in the forest during the dry season. Now, these results would suggest typical conservative isohydric behavior in the forest, with the species closing their stomata early to prevent water loss and sustaining a higher turgor loss point value as to compare to a more liberal anisohydrate behavior in the desert species, sustaining much lower water potentials at trigger loss point. But ANOVA results revealed no statistically significant differences between the trigger loss point means compared across these three ecosystems for both wet and dry season. So now I want you to shift your attention to the plot on the right. And here we're looking at the range of our T50 values stratified by biome. And we observe the widest range in the shrubland during the wet season and a substantially narrow range in the forest during the dry season. ANOVA results did reveal one statistically significant difference in the T50 means between the desert and the forest during the dry season. But it is worthy to note that with the exception of the forest, that the variation within the ecosystem is greater than the variation between ecosystems, which makes the use of heat tolerances to understand plant ecology complicated by the fact that tolerances can vary markedly among co-occurring species. Now, recent studies have shown that the interspecific variation of heat tolerances within communities often exceeds the variation in the community mean heat tolerances across coarse climatic gradients. So it would follow that heat tolerances should therefore correlate more with extreme leaf temperatures than with the regional climates. And this is especially since the leaf and the air temperatures can be decoupled. And this would suggest that leaf temperatures are determined by the physical properties of the leaves. And since plant species possess unique combinations of thermoregulatory traits, including leaf size and stomatal conductance and thermal absorptivity, different species should experience different leaf temperatures, even in identical environmental conditions. And so let's take a look at our next slide of data that I'd like to highlight. Our exciting result here are scatter plots of our trigger loss point on the y-axis versus our T50 on the x-axis. And we found significant p-values less than 0.05 in both the wet season and the dry seasons with our scatter plots using data from all ecosystems. The regression line reveals a strong negative correlation between these two parameters. And it is worthy to note that the p-value generated from plotting all the species, all ecosystems on our T50 versus trigger loss point scatter plots is more statistically significant than the p-values that were generated when stratifying the data by ecosystem. And that is because the p-value is driven by the higher sample size. So our major findings: number one, excluding the forest, interspecific variation with respect to drought and heat tolerant functional traits within a community of co-occurring species was greater than the variation between different communities across a coarse climatic gradient. And this finding would suggest that it is in fact not the ecological climate parameters that are driving drought and heat tolerance. Rather, it is the physiology of the organisms with respect to the functional traits for drought and heat tolerance that will allow species to acclimate and not succumb to climate change type of drought. Major finding number two, a significant negative correlation between our T50 and trigger loss point parameters emerged in both the wet and the dry seasons when we used the species data taken across all ecosystems. Now, these results suggest to us that drought and heat tolerant suites of traits could be conserved across all ecosystems, possibly as an evolutionary response to abiotic stress. And we posit that if the same suite of traits consistently recurs in diverse evolutionary lineages and across ecosystems with a broad range of climatic conditions, that it would seem likely that it evolved as a coherent unit selected for fitness rather than by parallel selection of each individual trait. And so therefore, our study linked the implications of drought and heat tolerance trait correlations to the trait co-selection hypothesis. And major finding number 3. So what is going on in the forest? So one possible hypothesis is that the co-selection for drought and heat tolerant traits occurred after the appearance of the gymnosperms in the phylogenetic tree, but simultaneous with the appearance of angiosperms. Now, I realize that I am speaking out of my area, but that is the beauty of ecophysiology, you see, because it can inform the formation of hypotheses scaling up to more ecological studies at regional scales, but it can also inform the formation of hypotheses scaling down to questions about the genome or phylogenetic applications. It is sandwiched there, right there in the middle. And so, in conclusion, it will be difficult to predict which species are most likely to survive the global change type of drought. But we believe our research opens the window to explore the evolutionary underpinnings that may lead to a phylogenetic signal for both drought and heat tolerance functional traits. Implications for understanding these evolutionary legacies, and coupled with genomic research, may progress to the identification of pleiotropic genes and could lead to improved predictive power to accurately identify the species that are likely able to survive amidst the forecasted combined heat and drought stress. I would be remiss if I did not acknowledge all of those listed here, but in particular, the American Association for the University of Women, Riverside East Rotary, and the UCR GSA for funding this research. And I'd like to thank everybody for your attention.